Today I am taking up the topic cell cycle and cell division, part 2, meiotic division. Meiotic division is also known as reduction division. Meiotic division results in the reduction of the chromosomes to half in the daughter cells. The meiotic division is exhibited by the gonadal cells of all sexually reproducing organisms. And it is generally seen in all higher plants and animals. The meiotic division has great significance. Then, the meiotic division is seen during the production of gametes, that is, male gametes and female gametes. The male and female gametes fuse to produce a zygote in the process of fertilization. If the chromosomal number is not reduced to half in the gametes, what happens? The resulting zygote will be carrying double the number of chromosomes. And if it continues, at every generation, the chromosomal number doubles up. But it has to be avoided. And this is avoided by adopting a cell division known as meiotic division or reduction division. Then, the meiotic division has also another great significance. During meiotic division, what happens is the homologous chromosomes exchange their genes, leading to recombination of genes. Recombination of genes lead to variation. Variation helps in evolution. And evolution results in the appearance of better species from the existing species. Then here we have to remember one more point. You take the human beings. We have 46 chromosomes in our somatic cells. Or we can say we have two sets of 23 chromosomes each. Each set is known as a genome. So, in our somatic cells, we have two genomes. Out of these two genomes, one genome or one set of 23 chromosomes come from mother and another set of 23 chromosomes come from father. They have different gene combinations. But here what happens is, during meiosis prophase 1, the homologous chromosomes of both the parents exchange genetic material. This is known as crossing over. How it happens, I will explain later on. Then, the meiotic division is divided into two stages, meiosis 1, meiosis 2. Meiosis 1 is entirely different from meiosis 2. Meiosis 2 is exactly like mitotic division. Now let us see how the meiosis 1 takes place. Then one more point is, at the end of meiotic division, 4 haploid cells are formed. During mitosis, what happens? At the end of the cell division, 2 cells are formed and they are diploid. But here in this case, 4 haploid cells are formed at the end of meiotic division. Then, coming to meiosis 1, it is divided into prophase 1, metaphase 1, anaphase 1 and telophase 1. First let us take up prophase 1, which is very, very important and crucial for genetic recombinations. The prophase 1 is also very long and it is subdivided into 5 phases, namely leptotene, zygotene, pachytene, diplotene and diakinesis. So in this way, the prophase 1 is divided into 5 subphases. Coming to the first subphase, leptotene. And one more point is, during entire prophase 1, the nuclear membrane remains intact and all the changes take place inside the nuclear membrane. Then, during leptotin, the chromosomes take up shape due to compaction of the chromatin material. So, the chromosomes slowly make their appearance. Then, in the next stage, zygote, the homologous chromosomes pair up. I have drawn a figure here, leptotin. I have drawn only one set of homologous chromosomes for the sake of convenience. So this is one set of homologous chromosomes. Let us say this is the paternal chromosome, this is the maternal chromosome. So during prophase 1, leptotin, the homologous chromosomes appear due to condensation of chromatin. Then in the next stage, that is zygote, the two homologous chromosomes, that is maternal and paternal chromosomes, come together and they pair up lengthwise, gene for gene. Remember, the homologous chromosomes have the same genes or their alleles in the same order. That is very, very important point. Otherwise, recombination is impossible. It leads to some other complications. So, these paternal and maternal homologous chromosomes have the same genes or their alleles in the same order. Then, the pairing of the homologous chromosomes is assisted by the formation of synaptonemal complex. So, synaptonemal complex holds the pairing of homologous chromosomes together. The next stage, packaging, it is very, very important. And it is much longer than leptotin and zygote stages. In the packaging, what happens? The chromatids of maternal and paternal chromosomes exchange exact segments between them. See, let us say this is the paternal chromosome, this is the maternal chromosome. Each chromosome has two chromatids. This chromosome has two chromatids, this two has two chromatids. Only one chromatid of each homologous chromosome participates in crossing over. So from this chromosome, this chromatid, and from this chromosome, this chromatid participates in crossing over. All the four chromatids do not participate. And before that, I want to tell you one more point. When both the homologous chromosomes pair up, it is known as dyad or bivalence. Now, the synapsal chromosomes are known as bivalent chromosomes. These two are known as bivalent chromosomes. Because there are four chromatids altogether, they are also known as a tetrad. Tetrad means four. So, when two homologous chromosomes pair up, we call them as bivalent chromosomes, and as they have four chromatids, they are also known as a tetrad. Out of these four chromatids, I have already told you, only one chromatid of each homologous chromosome participates in crossing. They cross like this and exchange exactly the same genes between them. As this is what happens, some genes present on the paternal chromosome are passed on to the maternal chromosome, and in exchange, some genes present on the maternal chromosome are passed on to the paternal chromosome. In this way, crossing over results in recombination of genes. Then after the crossing over, what happens? The homologous chromosomes try to get separated from each other. It happens during diplotin stage. The homologous chromosomes get separated, but still they are attached at the points where the crossing over has taken place. These crossing over places are known as chiasmata. They are X-shaped in structure and they are known as chiasmata. Then, after diplotin, diakinesis. And one more point is, during diplotin, what happens is, the synaptonemal complex disintegrates. Synaptonemal complex disintegrates. And one more point you have to remember. In crossing over, one enzyme participates to help in the process. That will do later on. Then during diakinesis, what happens? Both the homologous chromosomes get completely separated from each other. Now we can see the exchange segments of the chromatids. This red part comes from here and this black part comes from this chromosome. Similarly, this red portion comes from this chromosome and this black portion comes from this chromosome. In this way, exchange of genetic material has occurred between the homologous pair. And this process occurs among all the 23 pairs of homologous chromosomes. Then, diakinesis represents the end of prophase 1, which is very long. And by the end of the diakinesis, what happens? The nuclear membrane disappears. As this process is going on, the centrioles move to the opposite poles and they start producing spindle fibers. So at the end of prophase 1, what happens? Nuclear membrane disintegrates, spindle fibers are formed, and the chromosomes undergo still further condensation. Next stage is metaphase 1. In the metaphase 1, the chromosomes are arranged on the equatorial plane or metachronal plate like this. Here, there is one important point you have to remember. The homologous chromosomes are aligned like this. The spindle fiber of this centrosome attaches to this chromosome, and the spindle fiber of this aster belongs to attached to this chromosome. So in this way, during the metaphase, all the homologous chromosomes are arranged on the metachronal plane. One set on one side and other set on the other side. Then, anaphase. During anaphase, the homologous chromosomes are pulled towards the opposite poles. So one set of chromosomes move in this direction to this pole and other set moves to this direction. Here lies the important difference between mitosis and meiosis. During anaphase, in the case of mitosis, what happens? The chromosomes split into two daughter chromosomes. They split at the centromere. But in the case of meiosis, what happens? The entire homologous chromosome moves to the pole. There is no division of the chromosome into two daughter chromosomes at the centromere. The entire chromosome moves in this direction and this entire chromosome moves in this direction. As a result, what happens? The chromosomal number is reduced to half. This point you should never forget. So in the anaphase, what happens? In the meiosis, the entire chromosomes move towards the poles. As a result, one set of chromosomes reach this pole and one set of chromosomes reach this pole. Then, during anaphase 1, the chromosomes disperse into chromatin, nucleolus appears, nuclear membrane appears, and the cell is divided into two due to cytokinesis. In this way, at the end of meiosis 1, two haploid cells are formed. Then immediately the cell enters the meiosis 2. In meiosis 1, in meiosis 2, all the changes are just like mitosis division. So meiosis 2 is just like mitotic division. Here in the prophase 2, what happens is the nuclear membrane disintegrates, nucleolus disintegrates, and the chromosomes take up their shape. Then in the metaphase, the chromosomes appear on the metachronal plate or equatorial plane, and their kinetochores are attached to the spindle fibers. So each homologous chromosome is attached to the spindle fibers of either side or to the kinetochore of the centromere on either side. Then, during anaphase, the two chromatids get separated at the centromere. Then each daughter chromosome moves to the opposite poles. And it is followed by telophase 2. During telophase 2, what happens? The nuclear membrane reappears, nucleolus appears, the chromatin disperses, and 4 cells are formed due to cytokinesis. And these 4 cells carry only haploid number of chromosomes or n number of chromosomes. In this way, at the end of meiosis, 4 haploid cells are formed. I have drawn only few figures because of lack of space to explain the crucial points involved during meiosis 1, prophase 1. Here I want to explain about spermatogenesis and oogenesis, which is given in some other chapter. Spermatogenesis. In male human beings, what happens? In the gonads or testes, we find spermatogonia. The spermatogonia have diploid number of chromosomes, that is 2n number of chromosomes. The spermatogonia divide through mitosis and produce a large number of primary spermatocytes. The primary spermatocytes have also diploid number of chromosomes. These primary spermatocytes undergo meiotic division 1 and produce secondary spermatocytes. The secondary spermatocytes have haploid number of chromosomes, that is n number of chromosomes. The secondary spermatocytes undergo meiotic division 2 and produce 4 haploid cells having n number of chromosomes. These haploid cells are known as spermatids. So at the end of second meiotic division, 4 spermatids are formed. Then these 4 spermatids are transformed into sperms. Transformation of spermatid into sperm is known as spermiogenesis. The entire process is known as spermatogenesis. The last stage, that is transformation of spermatids into sperms, is known as spermiogenesis. Then coming to the oogenesis, which takes place in the ovary of the females. Oogonia are present in the embryonic ovary. Then the oogonia divide mitotically and produce primary oocytes. The primary oocytes have 2n number of chromosomes. That is diploid number of chromosomes. The primary oocytes undergo meiotic division 1. As a result, the chromosomal number is reduced to half and secondary oocytes are formed. Here is one difference. The secondary oocytes formed through meiosis 1 are not of equal size. One cell is much bigger, another one is very small. The bigger one is known as secondary oocyte and the smaller cell is known as first polar body. The first polar body serves no purpose. It cannot participate in fertilization. Then, both the secondary oocyte and first polar body divide through second meiosis. And ultimately, 4 cells are formed. When the secondary oocyte divides, again one polar body and ovum are formed. As the secondary oocyte is dividing, the first polar body also divides and produces 2 polar bodies. So in this way, at the end of meiosis 2, 3 polar bodies and 1 ovum are formed. Only the ovum can participate in fertilization and the polar bodies disintegrate. So in this way, spermatogenesis and oogenesis occurs in the human beings. Then, in meiosis, the duplication of DNA takes place only once, that is, during the S phase of resting stage before meiosis 1 begins. Then, meiotic division, I have already told you, is very important because it brings about recombinations and variations in the populations of a species. And these variations lead or they help in the natural selection, and due to the process of natural selection, new species arise from existing species. So that is about the meiotic division in plants and animals. Thank you.